It's a very bittersweet day. We're all here celebrating a life well lived and celebrating a person whom we all love. But we know it's a dark hour. But I'm not going to give up on Lucy or her mission. As you just heard, well in excess of 90% of the problems in cancer associated with metastatic disease, only 8% of people who apply for research dollars even mention the word, which is a very frightening statistic. And that means less than 5% of funding of research is going towards metastasis. And NFCR is one of the very few places that's had focus in that area. So I'd like to tell you where some of the monies that you have raised have gone and where some of it is going. My claim to fame, such as it is that we have discovered 8 metastasis suppressing genes that get turned off when cancers metastasize. We still are working on trying to understand how they work, but one of the amazing things is one of the genes, the first one we found, we discovered in Hershey when I was there, 14 years at Penn State. We are Penn State! Thank you. We called it KISS-1 to remind people it was discovered in Hershey. The second one we cloned is now known as BRIMS-1 because the genome people have no sense of humor because we'd initially named it HUG-1. But KISS-1 stops the cancer from growing after it's spread around the body. And one of the reasons I think Lucy found this project interesting is because that could mean we make metastasis a chronic disease like diabetes. We stop it in its tracks. We haven't eliminated it. We stop it in its tracks so that it doesn't cause problems with the liver or the brain or the lungs. And I would covet all of your prayers, good wishes, and so on. Next week, the culmination of 15 years' worth of work after discovering KISS is going to be an experiment that my entire lab from Shawnee, Kansas, I find it appropriate that we're here in Shawnee, Delaware, is going to perform. And it will involve 700 mice. And we are going to actually test whether KISS-1 will be a therapeutic. And if we're successful, and if it comes out as we predict, this is research, we cannot know for certain. This will be probably the impetus that pharmaceutical companies would want to push it forward. And the second project is one that NFCR has just begun funding, but I'm excited about it. And if you'll go back to junior high school, and you remember those things we call mitochondria? Remember those?
Try not to, Dan.
They're the parts of every cell that makes the nutrients that we take up and turns them into energy. What most people don't know is that mitochondria have their own DNA. And a colleague of mine and I had this wacky idea. No one, and I repeat, no one was willing to fund it. But NFCR allows me, with the grants from the Lucy Fund and from others, to do wild experiments. And what this experiment is to look at the DNA in the mitochondria and ask if that could control cancer. Nobody in their right mind would have necessarily known that. When you look at all the DNA in a cell, it's 3 billion bases of DNA. In mitochondria, it's 16,000. So 16,000 anything should not be controlling 3 billion. But guess what? mitochondria actually control breast cancer formation and metastasis. And it's single changes in the mitochondrial DNA that we've mapped, at least in one case, that suppress metastasis. This could mean we will be able to do a simple blood test, get the sequence of the mitochondria in under an hour, and plan somebody's therapy appropriately. So we're not there yet. We're not really that close. But that is something that you have funded, and we'll continue with that. So thank you. And now I'm going to just adhere to what are well known as the 3 B's of a good speech. Begin, be very brief, and then be seated. We all are in this together. Hold my lab, hold NFCR, hold any of the people you're funding accountable. You've worked hard to support us because what we hope we're bringing to you is hope. And with the indomitable spirit of Lucy, the whole family, and all the folks in our lab, we will continue fighting on until we end metastatic disease. Yeah. Thank you very much for coming.